Hi, Pushpinder. Good
Hi-
morning.
Hello, ma'am. Good morning.
Today we are going to discuss BLAST, which is a primary tool and a very simple tool people have used for at least decades now to analyze sequences, compare sequences, and decipher the biology, right? So, what I'm going to do today is to, start with instead of saying how to use BLAST, which is like very simple. Everybody can do it, and NCBI has done it so easy. You cut and paste your sequence, and it will give you some results. You like it or not, you have something. So it is not how to use BLAST that's really an important thing here. But since it's so easy to use, I fear, and I think all of us should fear, how not to use it, because you can interpret biology wrongly and you look bad in front of others because your interpretation is totally off. And so it's important to know how not to use BLAST. So, to know how not to use BLAST, a little bit of you know catch up on molecular biology, because BLAST is sequence and sequence is related to biology and genome and everything. So we'll switch quickly back to central dogma of molecular biology. You have the DNA. BLAST can do both DNA and protein, so it's important that we relate to central dogma of molecular biology. You have DNA,
hmm
which transcribes and then translates to make protein. Okay. So in that process, what happens is because of the degeneracy in the codon, like three triplets, multiple triplets of the nucleotides can code for same amino acid. That degeneracy becomes a really important aspect to know, understand, and appreciate, so that you understand when to use BLAST, when not to use BLAST, which category of BLAST, like BLAST and BLAST P, there are multiple things. They've
hmm
been engineered in a way that you don't use wrongly, but still it will work. It won't tell you you're using wrongly, okay? It will give you results. But it's up to us to understand not to do that, not to overinterpret if we didn't use the right BLAST subcategory. So,
True. I think I,
yeah, go ahead. Yeah.
yeah, true. I think I cannot agree more with you on this. I think BLAST is probably one of the most used, I would imagine, bioinformatics tool in the history. But I guess it's also one of the most misunderstood tool when it comes to its application. A lot of people talk about it is so easy to use. As you mentioned, just cut and paste a sequence there, put a click button, and you get results. Voila, it's out there on the screen. But then what exactly the result is telling you. As computational scientists, we always talk about garbage in, garbage out. It is one of the tools which can easily and very well fit that perfectly, that scenario. Like you can put in some sequence which can be anything, and it will give you something out for sure. I think the tool is meant to give you results, but are those even useful? Are those even interpretable? Like, do we actually make sense? And I think there's definitely a big lack in community to understand that where is the sense in the results you got, or where is just some garbage which you probably should just ignore and not even take it further. And that gap is something which is tremendous. And I think with this conversation we probably can try to fill that gap a bit and make people understand that how BLAST can help them, to interpret their results much better, and what are the use case scenario where you should not use BLAST, where you should use BLAST, and how should you interpret the result and what get the most out of your hits.
Yeah, thank you, Pushpender. That's pretty much use case scenario is where we will end this, and the use case scenario is really important because that's when it comes when not to use BLAST, which BLAST not to use for a particular interpretation. And so, having gone back to this central dogma of molecular biology, this DNA degeneracy. When I was looking at degeneracy, interestingly, I got some other idea and appreciation of natural selection. So I want to share that with you. It may be dragging us a little bit away from BLAST per se, but it'll be useful in understanding BLAST. Okay.
True.
Degeneracy is, whenever. First central dogma, and the first is replication. In replication, there are errors in DNA, nucleotides, there are errors, and nature tolerated a lot of errors, and it was able to tolerate many more errors, still have the reproductive capability, is because of degeneracy. Because many of the DNA mutations did not change the amino acids. Okay, that's great. Then all the amino acids should be exactly same. Then why do we need blast, right? We can just say that, oh, this is not ending. But at the same time, those mutations and a little bit of degeneracy in the amino acid, there is like groups of amino acid, hydrophobic, hydrophilic, and that kind of thing. So by changing the amino acid to the same type, it was able to tolerate and still create maybe different functions for the same protein. That way, nature had many more tools to, you know, to create functions and complex organisms like human and everything. That's one aspect of it. Third aspect of it is it was able to, take more mutation when it actually changed the function. Okay. And then we have different functions for the same fold within which we call paralog. So you have homologs. What does homolog do? Not changing the function, but they're different enough, so speciation happens. So let's not go into that. But I'm just saying that the degeneracy has created this wonderful world. Okay. And yeah, that's why blast is needed. Is that wonderful world? How do you interpret that wonderful world? That's why blast is important aspect to understand. Okay. In the degeneracy, one of the thing I in my classroom lecture, Pushpender, I've done more recently is it's hard to understand the devastation from degeneracy. Okay. If you take a 8 amino acid stretch, okay,
Hmm.
and ask the reverse question that how many DNAs, unique DNAs, can code for the exactly that 8 amino acid. It goes into tens of thousands
True, true.
just by multiplying degeneracy. So that gives us a feeling for, oh my God, so many DNAs can code for the same. And this is relevant to BLAST, okay, this particular aspect I'm saying. So you can, as an exercise, I'm not going to, as a lecture, I'm not going to go into Blackboard and give you a sequence and do that. But anyone who is listening to this dialogue can just write 8 amino acid and actually go with the degeneracy codon table and use the degeneracy to compute how many. There's too many. So now BLAST has to deal with that. And that is why how not to use BLAST comes in. Okay. So now this degeneracy, one thing I'm thinking and I think I want to share with you guys is, isn't that beautiful? That if this degeneracy wasn't there, then there wouldn't have been any speciation. There wouldn't have been complex organism. I'm like, even thinking about blast, we can appreciate natural selection so much, just because there is mutation. So replication, a perfect replication machinery with zero mutation, every replication, maybe nothing would have happened. There would have been no speciation, no function expansion, no application. We wouldn't need blast in that world. Correct?
True, true.
But
That's right. Yeah.
we live in a beautiful world. You want to say something about that, Push?
Yeah. I think, no, that's true. Like how this speciation evolves and how this degeneracy concept helps to, make this beautiful speciation effect and understand it. And at the same time, this also I feel like is something which makes the results of blast sometimes not rightly interpreted by people. Like they try to, I guess, over-interpret based on this homolog function that, oh, maybe this is the same, so I can assign a function to it just because it's a homolog, which might not always be the case. I think that's one case scenario where I'm trying to go for examples of where people, I think, misinterpret the blast results. Is like, let's say, if I if I'm doing an experiment and I find a gene and I assemble a genome of it, and I find out, oh, my blast shows me it's a kinase. But do you think, can I safely say my gene is a kinase just because it's similar to any kinase in the world? Do you think blast a homologous when I find to a kinase like blast, do you think is it safe for me to assume that the function would be a kinase, or do you think it's an overinterpretation?
Yeah, and that's a good one. This is at the protein level you're talking about, where it's a little bit safer to say that it may be kinase, but you got to go look at the full length, because that's what we are going to get into. That blast is a basic local alignment tool. So you can find the signature. That signature alone doesn't tell you that it's a complete kinase, right? So we'll get to that. We'll get to that point.
Yeah.
Okay, that's a beautiful one. But now going back to DNA, that's where most of the errors happen. Is one other example. This may be a little off, but I have to share it here. When you do next generation sequencing, everybody has heard about next generation sequencing even before they heard about BLAST,
Hmm.
right? So next
Yes.
generation sequencing, you've taken the mouse genome and you fragmented it and you sequenced it. You have 300 base pair sequences of mouse. Okay, you know that we use mouse as a as an animal for screening drugs. Okay, all the functions are preserved, all the proteins are preserved. We know that,
Hmm.
we better be happy with that, because we are using the drug that has been tested in mouse, right?
True,
So
true.
we are very similar, although we don't look alike at all. We're very similar in terms of protein sequences. But now I said that you have taken the DNA and you fragmented and you got, 10 million fragments from mouse. Okay. Then there is something called, I am going to find how many of those fragments are similar in human genome. Okay. It's less
Hmm.
than 3%.
Yes, true.
Okay, that again goes back to the degeneracy I was talking about. Like in DNA level, you completely lose similarity. Yet at the protein level, you maintain 80 to 90% similarity. And you function, our digestive system, our breathing systems, everything is similar. Biology is more or less similar, right? So these are the two things that at least should tell us that DNA blast is very different from protein blast, and you have to ask yourself, what do you want? What am I looking for? Given a sequence, here where the little bit application comes in, I'm given a DNA sequence. Okay, actually, Pushpender told me to start there this lecture.
I still have
Yeah,
that question out there. So I think
yeah. So you
we
you
can,
tell me, you tell me. Go
yeah,
ahead.
we can try a few different ways here. So I think I have like let's say I'm not a bioinformatician or I know nothing about this. And one day I'm roaming around somewhere on my one of my walks around, and I found some water sample in which I recover like a short DNA sequence. And I see that doesn't match anything obvious I know, by think. And I start suspecting maybe it's a novel species, you know. So, how do you think in this case I would use a blast to check whether it is truly novel or it is just something already known but poorly annotated? What should be my questions with this short sequence of DNA which I got from an a water body or something? And what should I never conclude from my results? So, how do you think we'll answer these questions?
Yeah. So it's one of the questions you can say that, okay, you have been given a sequence and you said you are not a assume you're not a I'm
Yes.
going to assume that I'm a bioinformatician. Oh my God, I got a sequence. I got to say something. Okay.
Yes.
That's a pressure I have because I'm a bioinformatician.
Yeah, because I spend money, so I want my answers
Yeah,
now. So,
yeah,
I
yeah.
see I
I have.
sequence it. So how
I'm
do you
forced to
think?
give. I'm
Yes,
forced to give you an answer. I don't feel forced to give an answer. I'm equally curious.
yes.
Equally curious because this came from some life form. I want to know what can we say. What can we say about it? So I'm equally curious. I'm not feeling pressured, but I'm anxious. Let's say I'm anxious to know what it is. So if you have a DNA, if you gave me a DNA sequence, then I would, of course, use blast N first, and
Okay.
not to annotate the DNA, not to say what function it is, but to just say which organism is it coming from. Remember, I said that DNA diverge very far, like mouse and human DNAs cannot be compared. Having said that, now you expect 100% or 98% identity of the DNA fragment that I got from Pushpender to doing blast 10. If I don't get 100% identity, if I get 80 to 90% identity, in the database, like something called, one of the things that we need to understand about BLAST is, which is hidden. All of us are very happy it is hidden because it's complex, right? NR database, human genome,
True, true.
species-wise databases, and all that. Usually, when you're running BLAST, the default is NR database. That's non-redundant DNA database. Non-redundant DNA database is everything that people have done so far. At least that's what you assume. That's what it's
Hmm.
meant for. And then NR protein database is all the protein that has been deposited. So, since you started with DNA, and we're going to do BLAST N, so DNA
Hmm.
to DNA comparison,
Yes.
because I want to see whether it's identical. If it's identical, then it's, the organism, the organism. You have assigned an organism, and I can go to Pushpinder and say, hey, I know it is C. elegans, or I know it's coming from E. coli, or something like
Okay.
that. Right? First answer I get if it is identical. So I run BLAST in against NR. In this case, you have to do NR database because that's where everything is there. So you're searching one sequence against everything, right? So, and
Yes.
then you find it, you got it, you can tell him, you feel good. Oh my God, I am able to give something to Pushpinder now. So, but at the same time, it is not. Okay, it's not. Then I go back to Pushpinder and say, hey, it looks like it's a novel organism. What do you think it can be? So I'm in a conversation mode with the biologist because I don't want to kill myself. And, that's what bioinformatics, it always engages in the dialogue with the biologist to give them the right answer. So, Pushpinder, what will be your answer when you didn't? When you said that I got novel one, what will be the reaction, and how would you guide me now?
I think how I take is that it's always exciting, I guess, if somebody says, oh, it's a novel species. Oh my God, I discovered something new. But I feel in BLAST, a no hit might not always be a novel species, correct?
Right,
And
right.
how do you think you will guide me through that process to make me realize that, oh, no, if it's no hit, I don't want to say it's a novel species. And how would you think a user should be able to make conclude from that step onwards?
Yeah, this is an important thing because in not only in biology, okay, anywhere, a negative result is not a negative result, right? Like if you don't have COVID, then it doesn't mean if COVID result came negative, it doesn't mean you don't have COVID. It is not detectable. So like
Yes.
every other thing, there's a detectability limit for every test.
Yes.
Blast also is very similar. Within the blast detectability thing, it's not detecting it either because that particular species is not yet deposited in the database. Okay, so that's the only way you won't find it. Okay,
True, true.
so usually that won't be the case, but if that is the case, you are in bad luck. Like you're thinking it is novel, but somebody is already working on it. And this is always the tricky thing for all of us. Like you're patenting something, you can check the patent database to see whether anybody else has patented it, but somebody is working on today, right, parallel to you, you will never
True.
know that. That's the only way it cannot be there in the NR database. So it's
Okay.
okay. You are at least, you are happy that you're working on something very novel, that someone may be working on it or someone may not be. So you're with the rest of the world. One example I have to give you, there was this person in my, he was my office mate back at Immunex when I was working, and we were like in a race for gene discovery.
Hmm.
That's where the novel thing comes up all the time, okay? Gene discovery. And he quickly said that, oh my God, I found a novel gene. Okay, you're going to patent it and some cytokine or something. Otherwise, nobody is interested in those days, okay?
Those
So
are druggable targets. They make
yeah.
money.
So we were in the race for cytokines and lymphokines and chemokines and all that. He said I found a novel one, and the patent of attorneys have been said, and they're all working on it and stuff like that. At that time, Blast, this was the early 1990s. Okay, so 91, Blast came 91. So it hasn't become a simple tool yet. Okay, so and then some other one person said, let me check with Blast and let me see whether it's really novel. And obviously, it wasn't novel at all. Okay, so you can make a fool out of yourself. So I'm
True.
saying this because this is a Blast, podcast, and I want to tell you that Blast really contributed a lot of people from spending a lot of time patenting something and making a fool out of themselves.
I'm
Okay.
sure.
That's the number one, right? And
Yeah. I think I have a few more questions on this same line. So I think you said I will use blast n as a scenario. Why do you think I cannot use blast x? What will that do to my result? That's my question number one. And my question number two, as we were going by, I'm not a bioinformatician, and you give me some results. How do you think, in a layman terms, you would like to explain me things like percentage identity, coverage, and e-value? I think those are the things which is the first thing which pops out when I put a sequence in the blast web page. So how do you think, as a user, I should interpret these three terms?
yeah, so we're just getting there. Thanks for asking the question, which makes me to continue to say what I've been saying. So blast, and we know we're going to use the organism. First, I have to say that for the same thing you gave me, if my question is to find which organism is it from, I
Hmm.
won't use the translation of that same DNA that you gave me. The reason for that is sometimes the protein level can be very identical, still they can come from a different species. Again,
Yes,
I'm going to
that's
go
true.
back to the
The
uh,
divergence
mouse,
theory.
and yeah,
Mhm.
because the DNA diverges. So you're only answering the question of is it from an organism or not. Okay, so you can't use blast P to answer the same thing. You can say that this is mouse. It
Yep.
could be from a human, but you got a fragment that's actually identical. It hasn't diverged at the protein level. So you cannot to identify organism, you have to do DNA. Okay, you did DNA, and now I'm saying you have to do DNA, you can do it. But now you're talking about kinases, remember? So
Hmm.
you could, now I'm saying, okay, it's novel. So, okay, novel species maybe, but what else can I do? I cannot tell the species, but what function can it do? Okay, so I can translate it. You were talking about BlastX, TBlastN, and other
Yes,
things.
yes.
Okay, so they're all there are only two tools. Okay, BlastP and BlastN in the
The
bottom.
broader sense.
Other
Yes,
things
yes.
are just helping you. If you have a DNA, you want to do protein level comparison, then you use T-blast. And if you have DNA to DNA, but you want to do the comparison at the protein, so that's like nuances a little bit. Nuances.
Hmm.
So I'm going to talk to you about blast P. Okay, blast P is either you give DNA and the tool generously translate that into protein because you don't want to be translating it. Translating is a bad thing because you get two to three translation. Yeah,
True, true.
forget. Let Blast do that. Okay. So anyway, it will translate and it will analyze it. So anyway, the protein is so I'm going to say that I'm not going to bother about that because then if you translate it, little bit of detour here. If you translate it, you're getting three frame translation. Okay,
Yes.
and and one exon can be here in the first translation frame, another exon can be here in the second translation frame, and all that you have to merge all of them, and you don't. It's a complex
It
thing,
comp yes,
complex
that's
DNA.
that sounds
Okay. So
complex.
that's the interpretation problem. So here you need to know that proteins are exonic, small DNA, they come together, not exactly in the same reading frame.
Yes.
All three reading frame can have different exons. So that is for a different lecture. But just be aware that translating into analyzing is not, you have to still do some homework there, okay? But suppose you got nice, it's short enough that you got one exon and you translated it and you're blasting it. What do you expect? What are you expecting now? You know that that species is not there in the NRDNA database. Okay. So you don't expect identity at the protein level
Protein
in the
level.
NR protein database, or maybe it will be identical, but still you cannot assign species. That's why I'm saying this. You cannot assign species to it. Okay. If it's identical, you know it has that function. Okay.
Yes.
And this protein doesn't evolve much. Maybe this protein is developmentally very critical. Those are kind of hand waving things I can do. Even though I'm not a biologist, I can say, hey, this hasn't evolved. So some proteins evolve far to get different functions. Some close to replication and translation and stuff. They are very conserved. So you can find a stretch of, say, 80 amino acid it's very similar to something else, and at the DNA level, they are not similar at all, then you know one thing you know, whatever function it's predicting, not only that function, and then you also know that that probably is very critical function for a life form. It is not something disposable, dispensable gene. It's very critical gene. Okay, so
Yes.
those are things you will interpret.
So I have
Mm,
a question on this is that I think that like we always talk about BLAST is a local alignment, right?
mm.
It's a local thing. So we get uh as we're talking about evolution and things like that, like when I make a hit to something, I see that my local alignment, the BLAST, is now going to a bacterial species, a plant species, a human species, and I'm getting all of them as my hit. So do you think uh, is it a good phylogenetic tool? Can I study how the evolution of this protein happened across speciations, or do you think it's something which I'm over interpreting my results when I look at multiple species as a hit for one small protein?
And this is again, I have to, because BLAST is a local alignment. One thing I'm going to, there are two things. First is BLAST is a very clever tool. People want AI, ML these days. BLAST has lived 30 years now, and I believe it will live immortal, I won't say, but it will live for next 20 years at least. The reason for that, the artificial intelligence, how we look at sequences, how as a human I'll compare sequences by writing in my notebook, has been captured, and the biology has been captured in his algorithm. Okay, so I may have time to say what exactly I mean by here. Okay, so the other thing is, when you're doing the when you have blast output, you're kind of comfortable with all the hits. That
Mhm.
means you brought the question, Pushpinder, of e-value, right? E-value.
Yes.
Yeah, I'm comfortable with the e-value. That also again is comes from experience. For protein, a particular e-value, you can get very comfortable. For DNA, you want to be very stringent about the e-value and stuff like that. We'll leave that alone because e-value is a good tool, but it also comes from experience. Sometimes you can say a probability of zero point 0.3 is good. Okay, 30% is good. Sometimes you say, no, 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 probability 90%. So it comes from
Okay.
what you're expecting, your biology, and what the odds of finding something. So I'm going to leave the e-value at this point. Okay, but you say, for example, if I have to tell you, you came with that, I say, e to the power minus 30 is good. Okay,
Why not 31?
if I not 31, or you can say that, okay, because you want to do phylogenetic tree, you have to have a cutoff somewhere. Okay,
Yes.
you can
Yes.
keep on going, or you could even, now I'm thinking loud here, you can take all of them and say that, okay, these e values are definitely not in this phylogenetic tree, less than e to the power minus 20. If it is not thousands of them, you cannot even decently do
Be-
phylogenetic tree. If there are, say, 70 or 80 of them, and you have really low, high E value, and you don't want to be deciding because you don't want to believe Subha ma'am when she says e to the power minus 30 or e to the power, I want to figure it out myself. You can do the phylogenetic tree. The ones that are very low E value, they will fall up away from the
Away
phylogenetic
from the.
tree and they will be totally away. Then you know that those e values are not real. Okay, then it falls in the nice phylogenetic tree. So there's n number of ways to do that. Okay, there's nothing etched in stone that this is the e value you need to believe. So I would say, yeah, you take. In fact, Blast allows you to click all the ones that you believe could be that family. You can select all of them, and you can do multiple sequence alignment and phylogenetic tree right there in Blast.
So it's a so I think I if I want to just make it in a short way, it's like it's a good stepping stone to give you what information you need, but you definitely need to start using much further things like multiple sequence alignment, things like that to make more sense of how evolution of this small thing might be going on.
Yeah.
Uh, I think since we are towards the end of what we thought our timeline would be, I have like few rapid questions for you. Do you think you
Okay.
wanna take
Okay.
that up? Let's try that out.
This is
Okay.
your turn to test me out, right? Okay, go
Yes, so
ahead.
they have to be, it has to be quick and crisp answers, which we think, which I think are, since I'm being a user here and you're being the expert who has seen Blast since kind of its inception. So, what do you think are the most common ways you have seen that students or researchers or big scientists, as you mentioned, they misuses Blast?
Okay. one of the things that I have seen my own students do, why I'm even doing this podcast, is because a biologist gives a sequence and say that, hey, I want to see whether this gene is there in all these other species. I've sequenced a lot of genomes of many species. I want to see where the homologs. Okay, then you take that cDNA of that gene, okay, and then you do blast N against other species. That's
Hmm.
complete garbage because now you are blasting DNA against other species. This is to hammer the point that you can never use blast N for giving the homology search because you will not find homology. But the crazy thing about it is you do get good hits at the DNA level. Now I'm going to answer you. You said rapid, so I'm going to stop right here.
Yes. My next is, so I think this is something which I think I personally always feel. Like I feel blast results a lot of time look very convincing. Like I think I feel like that. I think as you mentioned, your student did something and they found it looked very good. What do you think would be the place where one should be most skeptical or most, pay most attention to that output screen of blast?
Okay. As I tell my users sometime, is like if you are familiar with a protein, okay, here's where the familiarity comes. The analogy I use in my class all the time. There are there's 10,000 students, all girls who are all dressed in real uniforms and everything. But when you go there, okay, blast can't do that. Okay, it will find you. You're fair. Okay, hair done. All that blast can do, but blast cannot find your daughter. Okay,
True,
you
true.
will never take the wrong girl home.
Yes.
Okay, that is intelligence. Okay, in spite
So,
of Blast having done that intelligence. So so what I tell my, do you know you are looking for cytokines, right? Suppose you are looking for kinase. You just mentioned kinase. Kinase is a signature region in the protein. Those are called profile. We'll
profile.
cover them in a different lecture. Okay, so each protein, as they diverge, they still maintain regions of them because that is critical for kinase-like function. So there is a kinase motif. So if
Yes.
your blast, it's a local alignment. If blast found that motif, you can get excited about it. Then you don't even care E value, P value, whatever, whatever. Right? Then you can say, oh, maybe I got it. Now you're looking for another pair. Okay, if it is a kinase, there should be at the 200 amino acid level, there should be a profile. And I know that at the 75 amino acid level, there should be a profile. The reason why these two profile are maintained, I'm telling you now, is because they come together in the
In
3D
the 3D.
and
Yes.
important for function. And BLAST even in BLAST algorithm even inherits that because BLAST says that if I find only two regions, of words. I'm not going into algorithm, but I'm saying it's a word search algorithm.
Hmm.
It says that
Hmm.
I at least need two words
To
between my
match that
sequence and a match. Okay, the assumption is those two are probably coming together
in
and
the 3D
combining,
space.
right?
Yes.
So then you
True.
can be satisfied. Now you really found your kindness and you can go after that.
Yes. I think my last question, which I think would be like more of like a modern relevance to the world of AI, you already mentioned that. Like, as we know today, we have AI models, we have this massive database, we have much fast aligners. And why do you think BLAST still matters? And or where do you think will it slowly fade out of some way, or do you think it will still remain relevant for a very long time?
The reason why it will stay relevant for a very long time, it's going to be probably still the most efficient tool to get your first answer. Like, you give me a DNA, what can I do quickly?
Yes. Yes.
Still, it's going to be the most useful that way. Okay. And for AI and other thing like AlphaFold and other thing where sequence search, I believe, because I'm a structural bioinformatics basically in training, I believe that sequence search, which will be covered in a different podcast maybe, but structure comparison is the most meaningful thing in biology because sometimes sequence may not even be similar, but the structure will be
Structure.
similar.
Yes.
Okay,
Yes.
because of that, you will think that AlphaFold is going to take over. Okay, but still, It is not that efficient. Still, we have not gone that far. Still, we need to do something like BLAST need to come up for structure comparison in terms of efficiency, in terms of meaningfulness, in terms, and that is, I think, decades away. You may use after you got the kinase, right? You got your kinase.
Yes,
You could use now, go to structural bioinformatics and do AlphaFold to see how it folds. Are the
yes,
two profiles coming together?
true.
Is it going to all that you can do? But still, the initial traction you need to get from BLAST.
So it still will remain like a big thing, which is will be very useful. I'm sure as I think as we all talk about, it is definitely one of the best first steps to look into your sequence, make some sense out of it, make get some information, some first, I guess like a directionality tool is what I would call BLAST as, right? It gives you some direction of where you can further go with your sequence and find something out
Yeah,
of it. So I think that's a good takeaway point for people who are listening to this, that
yeah. I
it's
will tell
not
you.
going away anywhere.
Yeah, I will tell you that, well, it's very useful tool for biologists. I am not a biologist, and so I think that BLAST offers a very big way to get into biology. You understand biology. This is the tool that you can understand biology. You can dig. There's a database out there, there are sequences out there. How do you even understand biology through BLAST? Okay, that's also why I think the BLAST is going to live for a long time. Okay.
No,
So,
that sounds good. Yeah.
yeah.
So I think we have ran over 30 minutes now.
Over 30 minutes. So we'll end it
Yes.
now. Thank you, Pushpender. I hope
No,
I'm pretty sure listeners have many more questions, and hopefully
sure.
we'll
We
address
can,
those things in our consequent series. And so thank you so much.
no worries. We
Happy
can
holidays to
always
you. Bye.
do you too. Yeah. Thank you so much. Bye.
Bye.